Welcome back. In this video, I will discuss how pedigrees are constructed and analyzed. Mendel's experiments on pea plants provided valuable insight into the pattern of inheritance, such as how traits are passed on to the offspring. Not only that, but one can also predict the probability of a particular trait occurrence, whether the offspring will be tall or dwarf. However, studying genetic traits in humans present unique challenges. Experimental breeding and generating large numbers of offsprings are neither possible nor ethical in humans. To overcome these limitations, geneticists have developed alternative techniques specifically designed for studying human genetics. Three commonly used approaches for studying trait transmission in humans are pedigree analysis, twin studies, and adoption studies. In this video, we will focus on pedigree analysis. Pedigree analysis involves constructing a visual representation of family's history, often in the form of a family tree. This allows researchers to understand the pattern of inheritance for one or more characteristics within the family. Pedigree analysis is particularly useful for studying single gene disorders that adhere to Mendelian principles, assuming there are no complications such as incomplete penetrance or variable expressivity. By analyzing the pedigree, geneticists can assess the risk of passing of specific genetic traits or disorder within a family. This information is crucial for providing genetic counseling, making informed decisions regarding family planning, and guiding healthcare interventions. First, check the conventions used to construct a pedigree. In order to construct a pedigree, a geneticist must follow this convention. Some of the common conventions that are encountered frequently are discussed here. Look at this picture. Here a circle represents a female and a square represents a male individual. A diamond shape is used to represent if sex is undetermined. For affected person, it can be both male or female. The shape is filled in with red or black. An unshaded or empty shape represents an individual who is unaffected. The proband or the first affected family member coming to the attention of the geneticist is indicated with a solid color and a capital P with an arrow pointing to the shape from the left side. A diagonal line drawn through a shape indicates that the individual is dead. A horizontal line between a male and a female indicates a mating or marriage. Consanguinity or marriage between related people is indicated by two parallel horizontal lines drawn between the symbols. A vertical line descending from the mating line represents their biological children. Sheaves are placed in birth order from left to right and are labeled with Arabic numbers. For identical twins, an extra line is drawn between these lines, resulting in a shape that looks like a big A. These symbols represent non-identical twins. Roman numbers are often used to designate each generation, with the oldest generation at the top. The numbering system is required to identify a particular person very easily from the pedigree. For example, if I say 3-2, it means this person. If 2-1, it means this person. Now let's check how a pedigree is constructed step by step. The first step is to collect detailed information about the family members, their relationship, and any known traits or phenotypes of interest. This information can be obtained through interviews, medical records, and family history. Using the gathered information, a pedigree is constructed. The pedigree diagram illustrates the family relationship and indicates symbols to present individuals, their gender, and their affected or unaffected status for the trait or condition of interest. The pedigree is carefully examined to identify any pattern of inheritance. This involves looking for consistent transmission of the trait through generation, observing the affected and unaffected individuals, and considering the gender distribution of affected individuals. Finally, based on the observed patterns, the mode of inheritance is determined. It allows for predictions to be made about the likelihood or risk or option or potential outcomes associated with the trait of future generation. There are several types of pedigrees commonly used in human genetics, each serving a specific purpose. Here are some of the commonly used types. Autosomal pedigree. This type of pedigree is used to analyze the inheritance of traits or disorder that are located on autosomal chromosome. It tracks the transmission of the trait or disorder through both male and female individuals in the family. It can be two types, dominant and recessive. If it is an autosomal recessive trait, you will find these characteristics. It is usually appeared in both sexes with equal frequency. The characteristics is generally observed in all type of autosomal traits but not in sex-linked traits. So when you find that both male and females are affected, that means it is an autosomal trait for sure. Trends to skip generation. Here affected persons are found in this and this generation only. Here no affected individuals appeared. Remember, skipping a generation is a characteristic of a recessive trait. Affected offspring are usually born to unaffected parents. When both parents are heterozygous, approximately 1/4 of the offspring will be affected. It appears more frequently among the children of consanguineous marriage. Autosomal dominant trait usually appears in both sexes and with equal frequency. Both sexes transmitted the trait to their offspring and it doesn't skip generation. Affected offsprings must have an affected parents unless they possess a new mutation. When one parent is affected, that means heterozygous, and the other parent is unaffected, approximately half of the offspring will be affected. Unaffected parents do not transmit the trait. Now I will discuss sex-linked pedigrees. It can be either X-linked or Y-linked. X-linked pedigrees specifically focus on traits or disorder that are located on the X chromosome. The X-linked traits exhibit different inheritance patterns in males and females due to the presence of only one X chromosome in male. It also can be two types, dominant and recessive. In a X-linked recessive trait, usually more males than females are affected. Affected sons are usually born to unaffected mothers, thus the trait skips generations. Approximately half of a carrier, that is heterozygous mother's sons, are affected. Never pass from father to son. All daughters of affected fathers are carriers. X-linked dominant trait, both males and females are usually affected. Often more females than males are affected. It doesn't skip generations. Affected son must have an affected mother. Affected daughters must have either an affected mother or an affected father. Affected fathers pass the trait to all their daughters. Affected mother, if heterozygous, pass the trait to half of their son and half of their daughters. Y-linked pedigrees are used to study traits or disorders that are located on the Y chromosome. Since the Y chromosome contains very few genes and is only present in males, Y-linked traits are passed exclusively from father to son. Only males are affected here and it passes from father to all his sons, and it doesn't skip generations. Pedigree analysis has significant importance across different fields, including clinical genetics. Pedigree analysis aids in identifying affected individuals, determining the mode of inheritance, and predicting the risk of passing on the condition to the future generation. And in population genetics, pedigree analysis is used to study the inheritance of genetic traits and their frequencies within a population. Pedigree analysis is extensively used in animal breeding and agriculture to assess the inheritance of desired traits, such as disease resistance, productivity, and specific phenotypic characteristics. So that's all for now. If you like this content, press the like button and leave your valuable comments. Subscribe to my channel and press the bell icon for regular updates. And finally, thanks for watching.